All right. Good afternoon, everybody. Let's welcome Dr. Bethany Edwards. Is a warm welcome. She's a professor at UC Berkeley and a seagoing oceanographer. She's deeply interested in how marine microbes interact with one another through chemical signaling and their impact this has on the ocean biogeochemistry. Her lab specializes in lipidomics, which allows us to measure chemotaxonomic lipids, lipid-derived chemical signals, and energy storage compounds, as well as biomarkers for various types of stress. They are particularly interested in integrating across omic platforms, pairing lipidomics with metagenomics and metatranscriptomics to tell biogeochemical stories. So I'll hand it over to you now.
Well, thank you so much, Lucas. Well, I've had a really wonderful time visiting today. This is my first time in almost 8 years at Berkeley, 7 years at Berkeley, visiting Bodega. So I'm sorry it took me so long to get out here, but I hope to be back soon. And I've been really impressed with the facility y'all have here. So I'll give you a quick outline of where I'm going with my talk today. I'll start by reviewing the biological carbon pump. All the organisms that I'm going to be talking about today kind of integrate into the biological carbon pump. I'll also give you a brief introduction to lipidomics, how it fits inside of our view of transcriptomics and genomics. Oh, thank you so much. So, I'll give you some examples of some laboratory lipidomic studies. Looking at chemical signaling from diatoms whenever they're under and growth phases, and also and I'll talk a lot about diatoms, stress, the chemicals that they laboratory experiments, and also biological carbon pump. We have phytoplankton in the surface ocean, right? They're fixing carbon, they're taking CO2 out of the water, which causes atmosphere and drives CO2. Phytoplankton fix that into their biomass. Phytoplankton can die and sink particles, but they can also up the trophic food web. Until probably the early 2000s, most people thought of the canonical phytoplankton like diatoms going into copepods and then copepods going into fish. And certainly this pathway exists, but we now recognize that microzooplankton grazes like dinoflagellates, radiolarians, forams, they play a part in biomass and surfaces. Also have viruses and phages. Phages as by this process, bacteria in particles, or could be sinking particles, suspended particles. It's released into this dissolving carbon that would have been sinking through the water column as particles, but it's now less traction, but we're exotic phytoplankton viruses. Viruses infect. Infection causes aggregation, and sometimes it causes the production of things like transparent exopolymers, sustainable particles, kind of this mucus aggregation and enhance. So these are, all the organisms. Also transporting that carbon into the deep ocean with them, where they'll live part of their life stage up in the surface ocean, and then pods have their reproductive cycles key to this as well. And so they'll either produce eggs in the surface or at depth, functions to move carbon into the deep ocean. Phytoplankton in the surface ocean, something like 5 to 500 meters. And the reason is thousands of years. There's a really big role in our natural. How this relates, that we produce something like carbon per year in fossil fuels. So about pumping carbon from the surface. By vision of 5 years. So about 62% of that fixed biomass. So these microzooplankton do a lot of work. I put an asterisk by the 6% value of mortality going into viruses because these are early numbers. There's some studies that suggest viral infection is really hard to measure in the environment, and so there's some mortality due to viruses could equal or be equal to mortality due to grazing in certain systems. Let me just fix my mic here. So this value may be a lot higher than we know it to be. It's a low end mortality is actually caused by viruses. Quite a few papers looking at this across latitudes and longitudes. Yeah. Other side. Since I'm looking that way. Smart. I like it. There we go. Okay. Hopefully that works a little bit better. So other people have thought about the biological carbon pump in this way. We typically roll all of the information I just gave you in different pumps. Biological gravitational pump, this is just like the sinking of particles. Particles sink out 1 to 100 meters slowly into the deep. I mentioned some of the activity of zooplankton. They're moving carbon from the surface ocean to depth hours. Really nice review paper by Boyd. Bring these processes up because we're just now starting to constrain them. Biological carbon pump over time. These other processes again. add up all of these other processes other than just all these interactions likely impact. I'll give you a little bit of background on lipidomics. I wrote a review paper and I attempted to put the field of lipidomics in the future. Here wave of lipidomics. But we've been looking at biomolecules. These are some figures that I recreated from work with Jim's group. Ox-era oceanography on measuring fluxes. 80s, you can see that acids, carbohydrates. At the time, these were just measured. And later, and even back in the 70s, though, we were basically take a lipid sample and break it down. Hot material versus suspended. Thinking particles are very different from transformation. Give you a sense of, we've been doing this type of lipidomics, but we've been at this revolution. Give you some idea of what is a lipidome. Our pipeline looks like. Higher extraction, so phytoplankton on it. And then we do an organic extraction where we're adding organic solvents and water, and then we add more solvent at the end. All the water-soluble molecules. We separate our molecules based on measure those masses and kind of I had something that was 15 and now I have 7. How do I get 7? Of all of these spectral libraries that we search for. Carbons and 8 double bonds. Backbone, fatty acids. we're able to know that this is exactly how this fragments, often what they weigh. I would spend a ton of time on this piece. It's aspects of high performance liquid chromatography. Really accurate masses, so like to make that. Now I can take my coat off. Need the pocket. And so we get our data off of our instrument, and then we have a couple of different pathways that we can send it down. We tend to use this LOPSTAS database that I helped create when I was in graduate school. LOPSTAS is an acronym. It stands for Lipid Oxidation Biosignatures Triglycerides Adduct Hierarchy System, I believe is what it stands for. But essentially what we're doing is we have like an in silico database that has like all the different lipids that you could potentially have, and we're querying against that. So it's untargeted in the sense that in the data I have everything, but then on the back end I pick very specifically by using certain databases. And then once we have our like putative annotations, we go back and cross-check with other programs that allow us to look at fragmentation patterns, predicted fragmentation patterns, networks of fragmentation patterns from other data, and then we can get an annotated lipidome. And so this might be something like 10,000 features that you're able to reduce to like 1,000 compounds. And just to give you some like scale. And then we use a bunch of different statistical packages to do things like multivariate analysis, analysis of variance, and also try to take some of that metadata. So information on temperature, growth conditions, nutrients, that kind of stuff, to see if we can work out exactly what's going on in our system. So this is, our general workflow. I mentioned the blind Dyer for particulate lipidomes. We also look at dissolved lipids, so just whatever's out there in dissolved phase, meaning we've removed the particles. So compounds that are available either for take-up by bacteria or potentially that are available out there for chemical signaling. So this is the lipidome of Phaeodactylum tricornutum. This is kind of a lab rat diatom. It was one of the first diatoms to have its genome sequenced. And so here we have time. So this is a chromatogram. So earlier on our timeline, we're getting things that are more water soluble. Later on in time, we're getting compounds that are more hydrophobic. And so we end up with structural lipids, intact polar lipids that make up cell membranes. That's probably where you've most thought about lipids outside of like dietary constraints, right? So this is like the polar head group that loves water, and then like the two fatty acid tails that do not. And then we also have within the lipid realm energy harvesting lipids. So these are pigments and quinones. So if you're a photosynthesizer, you're going to have pigments. Quinones are used by everybody to shuttle electrons across membranes during energy acquisition. And then down here we have the triglycerides. These are energy storage molecules. When we talk about fats and foods, we're normally talking about these types of compounds. And then lastly, we have our signals. They're more water soluble because they tend to be smaller and have oxygens on them. So we think of like lipids don't dissolve in water. These ones kind of do because they've been modified a little bit to make them more soluble. Today I'm going to tell you stories just about
signaling lipids. But if you're interested in these other types of lipids, my graduate student, Jiwoon Wang, she had a paper earlier this year looking at diol cycles in all these different subclasses of lipids. And from the pigments, she was able to work out community composition, and we sampled over 5 days and saw pretty stable community that on day 3 that co-occurred with a new moon and an increased tidal influence at our site. We saw this like shift in the community composition. And we've since been able to do some cross comparison with DNA data. And it seems like our lipidome, pretty well captured what was going on. Clearly with DNA data, you're gonna get much higher resolution taxonomic data. But we were still able to catch that there was a big decrease in the diatom biomass, and haptophytes and Synechococcus tended to grow up over time. And the DNA data that we've looked at says that pretty well. So if you're interested in these types of chemotaxonomic lipids, as well as some of these triglycerides, I would suggest this paper. But today we're just gonna focus on signaling molecules. I'm gonna check the time because I've given you quite the review here. So let's jump into some data. We'll talk about some lipidomic screenings and what oxylipins are to begin with. So oxylipins are fatty acids for membrane lipids. So here we have nice membrane bilayer. Here we have those structural lipids that make up this membrane bilayer. And this blue one here is EPA. And this molecule will get cleaved from the intact polar lipid. And then once it's cleaved, you can add hydroperoxy groups here by this enzyme lipoxygenase. This enzyme can act on any position, any of these positions on the backbone. So carbon 1, 2, 3, 4, 5, 6, 7, so on, so forth to 20. It can act on the 5th carbon, the 9th, 11th, 14th, 12th, any of those. And it can act on any of these fatty acids with more than 2 double bonds. And so you can see that there's just a lot of iterations of oxylipins that you could potentially make. But once you've added this hydroperoxy group, that's an oxylipin, and that compound can be used in chemical signaling. And then there's other modifications, and these are all oxidized lipids. So what do these lipids do? Who are they produced by? Who makes them? They tend to be produced by diatoms. Diatoms have a lot of this compound here, EPA. They also have a fair amount of DHA, and these other polyunsaturated fatty acids are really common in diatoms. They can be produced under abiotic stress, but also during grazing by copepods and by these smaller flagellate grazers. And all these circled ones are interactions I'll talk about today, but they tend to inhibit other eukaryotic phytoplankton. They tend to inhibit microzooplankton grazing, and they inhibit the viability of copepod eggs. So copepods are less responsive grazing-wise to these compounds, but it will cause mutations in the nuclei and cause as much as like an 80% decrease in cell viability when copepods are given things that only produce these oxylipins. And so they're allelopathic, meaning that they impact the grazing rate, the growth rate and behavior of other compounds in a negative way. And they're clearly used as chemical signals that kind of like radiate throughout these surface ocean communities. So we're interested in what happens whenever you infect diatoms with viruses. So we knew from the literature that if you take some diatoms and you sonicate them, you'll get production of these oxylipins. If you take some diatoms and you expose them to really high temperature, really high UV, they'll produce these. But what happens under like more natural circumstances? And so we tried this with 3 different diatom host virus pairs. So we had Cattoceras tenuissimus infected with a DNA virus, and then the same host infected with an RNA virus. And then we had Cattoceras socialis infected with an RNA virus. And so we have a couple of different types of viruses here, DNA viruses versus the RNA viruses. They have different pathways of infection. If you're a DNA virus, you gotta express your RNA to actually start making genes to like do something during infection, versus RNA viruses, you already have like an mRNA transcript essentially whenever you right? And so then you can start making a virus, can start making copies of itself and the machinery it needs to undertake viral infection very, very fast. So we compared just the host during its normal growth phase. And then we also tried the host whenever it was infected. And we sampled early on in infection, like 1 day after infection, a few days later, and then as we saw the cells start to crash, we also sampled. And then we sampled the controls, just our host by themselves, at the same times that we sampled for viral infection. And then we let them grow a little bit longer and sampled them like as they're starting to decline, like 30 days later or so. And so we had a better comparison. This is just looking at all of just adding up the peak area. So this is like a proxy for concentration. It's like instrument signal across all of our different treatments. And we saw that there was a little bit of a response in the controls, which are in blue. During early stationary phase, you get a little bit more peak area. But during infection, even on day 1, they're already starting to leak a bunch of fatty acids and oxylipins into the media. Even though cell concentrations haven't decreased yet, we can already see the response in the dissolved metabolome of the system. So this is all the compounds. We saw 256 different compounds that we're able to identify. And red means they're higher relative abundance. Blue means lower relative abundance. We're giving equal weight to high abundance and low abundance compounds. So these are all relative to themselves. And so we end up with like 2 clusters of samples. These over here are either really, really early infection in the pink, or our controls in the blue shades. This yellow is our decline samples. And then over here we have mostly things that were either just transferred on transfer day, or sorry, not transfer day. One of our Catastris socialis in early stationary phase was making a bunch of things. But the rest of these samples are viral infection. And then this guy over here, this group of samples is our decline from a non-host diatom. And so the thing I want you to take away from this is that clearly there's a big response with viral infection compared to our controls. And we're seeing like a big response in the dissolved lipidome. There's a lot of lipids being produced. And another takeaway is that when we were looking at Catastris socialis versus a different diatom, Catastris tenuissimus, it seems that there can be a lot of variability at the genus level on what's produced and when it's produced. So this one outlier that wasn't really in our experimental scheme showed that this particular Catastris could produce a ton of oxylipins in stationary phase as well. So this complicates our view maybe of using these as biomarkers for viral infection, but they still gives us a good insight into whenever we're thinking about what happens as blooms in the natural environment kick up and either get infected or get nutrient limited. So to dive a little bit more into this, we analyzed our hosts. We had one host that was infected by 2 different viruses, and we had 2 different hosts infected by similar types of viruses. And so just comparing, maybe for right now, I won't talk about the different host differences. I already mentioned that Catastris socialis seems to be doing something a lot different. It would suggest that like in the environment, if you have Catastris socialis, it's going to be kicking out a lot of essential fatty acids that could be taken up by other organisms, right? Versus Catastris tenuissimus, which wasn't. But we saw a big difference in Catastris tenuissimus when it was infected with DNA virus versus RNA virus. We saw that, so here we're comparing all the compounds that were upregulated during lysis of the DNA virus, and then all the compounds that were upregulated during lysis of the RNA virus. We're comparing the number of carbons in these compounds, the number of double bonds, and the number of oxidations. And so the RNA virus, as it infects, it leads to longer chain length fatty acids and also more oxidized fatty acids. This is actually telling us something about the subcellular progress of infection. So Lipids tend to be compartmentalized inside of organelles within eukaryotes. And this is actually one of the lines of evidence for endosymbiotic theory, is that when you look at organelles, you can see the ancient lipids of the cyanobacteria that was eventually became the mitochondria, that became the chloroplast in the mitochondria that was once a heterotrophic bacteria. And so what this tells us is that during infection with RNA viruses, we're getting more oxidation of endoplasmic reticulum, and we're getting liberation of lipids that are only really found kind of like in this body. Versus during DNA infection, we ended up with a lot more lipids that are favored by the chloroplast. And something we know from experiments with a bunch of metazoans is that when you add these chemical signals in, if you add longer chain link ones, they tend to be more toxic. And so there's probably some difference in the chemical signaling field and how the community is responding, whether or not you're infecting with a DNA virus versus an RNA virus. So that was one cool thing that came out of this work, is that viral infection does increase the production of these compounds, and then the type of virus and the type of host you have really matters when it comes to thinking about these types of things. So I mentioned earlier that these compounds can inhibit microzooplankton grazing. This is an example from a study that we did with Matt Johnson at Woods Hole. And so we took some of these compounds that we're seeing produced, and we measured how they were produced during viral infections. This is data I just showed you, control versus viral infection. And this is a relative fold change compared to the control, so we can put these both in the same category. And then this is a different experiment where we took Phaeodactylum tricornutum, one of these lab rats, and fed it to another lab rat, Oxyrus marina, which is a dinoflagellate, and we saw that this also increased the production of these compounds. But interestingly, we can buy these compounds or analogs of these compounds. So we bought 2 analogs of these compounds. They weigh the exact same, so they have the same number of carbons, hydrogens, oxygens. It's just that here, the 2 extra oxygens, the hydroperoxy group is down here on the 15th carbon. And over here, you have 1 hydroperoxy group on the 12th and 1 on the 5th carbon. So they have the same backbone. They're akin to EPA. But we saw that whenever we added these compounds, so this is the concentration here of the compound, and then here's the ingestion rate. So how many prey are being eaten per predator per day. And you see that the ingestion rate for the black compound, leukotriene B4, this one, falls off really quickly and stays really low. Versus whenever we add this other compound in, the community, it does decrease the ingestion rate, but not as severely. And the community seems to have a much higher tolerance. Either way, we're decreasing growth rate, but the community is having a differential effect based on which of these compounds, who are incredibly similar to each other, is there. I have a student right now who's working on looking at some of these species-specific responses to stress. So he's taken 5 different species of diatoms and grown them under silica stress and silica replete conditions, and sampled in stationary and log phase. For the sake of time, and because I want to get to some oyster larvae stuff, I won't talk about it too much. But essentially, one of the things we found when we looked at this particular suite of diatoms, looking at Thalassia rotula, which is known to make a lot of oxylichens in the Mediterranean, and then Catostris, this is a species that we got from the San Pedro Ocean Time series. Skeletonema japonicum is akin to another organism, Skeletonema cristatum, which is known to make a lot of these compounds in the Mediterranean, and then Pseudonychia multiseries. Pseudonychia known to be a harmful algal bloom species, although multiseries not as toxic. So we looked at all of these, and the major takeaway was that they all had a different response in their dissolved lipidome depending on whether or not we sampled them in log phase or in stationary phase. But most of them responded to silica stress in some capacity. Our largest response was from Pseudonychia multiseries, but Skeletonema japonicum was also a really big responder, as well as this other Catopteris species. This is just looking at the relative abundance of production. And so here you can see in log phase with silica and then without silica. So whenever this cell is silica stressed, it's leaking a lot more of these oxidized compounds. And these are compounds that are going to be chemical signals in the environment, right? We have some hypotheses about what exactly is happening here. We think it's these diatoms, they have the silica frustral, right? And so that gets really thin. And so we think that thinning is leading to more exudation and cellular material. There's also this process called blebbing that I haven't studied in much detail, but you can see it in lots of other phytoplankton where, especially during like apoptosis, as cells undergo cell death, they'll start to bleb off bits of their membrane. And so it's literally like a little micelle just like popping off and floating away. And there's a whole field of research done on that and like what's the cargo of these blebs. Sometimes they're called vesicles. Sometimes they can carry DNA. They clearly are carrying organic material. But I won't go into that too much, other than to say Skeletonema japonicum and Pseudonychia, those were our main producers. And so we did some experiments with grazers, and this is really preliminary work done by Hoshu Zhang at Woods Hole, where he looked at the swimming dynamics of a heterotrophic dinoflagellate called Gyrodinium spirale, and looked at it in fresh growth media, and then we compared that to growth media from some of our big oxylifen producers. So, oops, I have to start it over here. So Hoshu is working on like pulling out the numbers from this, but I think you can see the difference right away. So this is happy spirale swimming pretty straight lines, changing direction every now and then, versus the spirale that's been exposed to oxylipins from Skeletonema. You can see they look a little drunk, right? They're swimming around, changing direction, changing direction, changing direction. And so we're looking into this in a more quantitative way to get at turn speed, turn frequency. But there are hypotheses in flagellate grazing world that being able to swim a straight line is important for finding prey. And so clearly these chemical signals are impacting that. Implications from field studies. So I've showed you a bunch of laboratory experiments where we infect diatoms with viruses, they produce these compounds, we stress them out and grow them in different growth phases. And depending on which species of diatom you're looking at, they'll have a slightly different response, but we can certainly induce the production of these by altering abiotic stress. Let's look in the field. Do we see it out in the environment? And so we went on a cruise. These are data from my thesis, and I've reworked them. And if you go and read my thesis, you'll see this data presented in a totally different fashion with pieces missing, but that's the way it is sometimes. On this cruise, we occupied a number of different sites. So we had some sites down here, the crude geology here, it's due to the resolution of the model, but down here in Monterey Bay, and then up here in Point Reyes, and then a couple of offshore sites. These indicate when we sampled those sites, and this is just satellite data estimating primary productivity in the region. So the more green the value, the higher the primary productivity. So we sampled Monterey Bay during the peak of a bloom, and so you can see that primary productivity in this box, in this box down here, dips off after we were there sampling. And then we came up here and we sampled Point Reyes as it was crashing, so as this bloom was crashing. We sampled for diatom viruses. And we looked at cell-associated viruses, so viruses that we could pull out of the transcriptome. And then we also looked at extracellular viruses, so viruses that we could amplify from filtered seawater. And all the blue dots are samples that were from Monterey Bay, and all the red dots are samples that were from Point Reyes. And so in a paper that I have not cited here, Kranzler et al., we make some conjectures about having sampled Monterey Bay during active infection, where you have a lot of virus inside of the cell, but you also have a lot of virus outside of the cell. So you're kind of in the throes of infection. And we sampled Point Reyes during this like post-lysis event, where we have a lot of extracellular virus, but not a lot of transcription within the environment, or sorry, within the actual cells. And you know, that's corroborated by the satellite net primary productivity. Point Reyes has clearly undergone some sort of bloom crash. We put sediment traps out there. And these were sediment traps that were a little bit rudimentary. They're mostly for just collecting material to do experiments on. So they were placed at 50 meters for about 8 hours. And here we're looking at the flux of carbon milligrams per meter squared per day, and then nitrogen multiplied by Redfield, just to make everything fit on one little line here. And then the blue values are this E ratio, so it's comparing export to how much primary productivity there was to look at how efficient is that carbon export. And these values are a little bit low, but we think this is due to our sampling method, this like rudimentary sediment trap that we had. But they're still intercomparable. And one of the things to note is that on the last day of sampling, on July 4th, as this bloom is crashing, we observed the highest carbon export compared to these actively infected experiments. So this would suggest to us that we have some stimulation of this viral shuttle, so this export of material to depth during viral infection or post-viral infection, post-lysis. We get this enhancement in carbon export, even though primary productivity is really, really low. So we also get this jump in export efficiency. We're getting very efficient carbon export to depth. And whenever we look at the lipids across this sampling site, we saw the highest values were at our post-lytic site. And then the next highest values were in our active infection site. There's about 200 or so oxylipins that we observed. We can use multivariate analysis to kind of like group these apart and then ask which compounds contributed to this separation. And so up here, these are all the compounds that loaded positively on component 1, and that happened to be really high in our post-lytic site and really low concentrations in our active infection site. And the compounds here that I've highlighted in bold, these are actually compounds that we observed in our culture experiments with those catastrophes, the ones I first showed you. So when we're infecting viruses in the lab, we see the production of these compounds. And then whenever we look out in the environment, they're most associated with the highest values in this post-lytic site. So we've observed something in the lab, and then we've looked in the environment, and we see these same lipid biomarkers being produced during infection at our site. We also did a bunch of experiments where we took different communities. So here we have microzooplankton grazers looking at phytoplankton growth of their prey, and then grazing of the microzooplankton, and we've added in some of these compounds exogenously to see what would happen with, you know, real natural communities, right? Not these lab rats that have been in culture for 10, 20, 30 years. And for most of our sites, we see a decrease in growth rate and phytoplankton grazing whenever we add oxylipins. The places where this didn't hold true here, this non-blooming site, we had super variable results to begin with. So we probably had a pretty heterogeneous community that just wasn't really responding cohesively across our 3 bottles. And then we did some experiments where we took surface ocean communities, really trying to focus more on like bacteria. And we added different concentrations of these compounds. And in laboratory experiments, what we had seen was that if you take a bacteria that was just isolated from the surface ocean, and you add these oxylipins, what would typically happen is their growth would be inhibited. And there's this big like screening study, and they look at like 30 some odd isolates, and almost all of them were inhibited, and there was only 3 that weren't. And the 3 that weren't were ones that were isolates that had been isolated from bacteria, or sorry, from diatoms. So they're bacteria that live in association with diatoms. Here, we've gone out into a diatom-dominated community and found that the bacteria weren't inhibited. Their enzyme activity actually was stimulated by addition of these compounds with respect to peptidase. And with respect to phosphatase, looks like we had some, stimulation at 1 micromolar, but once we got to 10 micromolar, we're still getting, lots of peptidase activity, but really low phosphatase activity. Versus when we looked at particle-associated communities, so these are communities that we've gotten from these sediment traps at 50 meters, versus like these are communities from like 5 meters. Whenever we added like the same concentrations of oxylipins, we get stimulation, but we get stimulation of a different enzyme. So we get stimulation of phosphatase on particles and stimulation of peptidase in surface ocean communities. So just again showing that we have this ubiquitous signaling molecule that's being produced by diatoms, and it's affecting different components of the microbial community in vastly different ways. The difference between these two sides is different compounds that we added. We tried adding a bunch of these really long compounds that we had played around with in laboratory experiments. We didn't have enough of it at sea at the time to show too much, but in one experiment at least we did get, or 2 experiments I guess, we did get stimulation that kind of matched what was going on with these other compounds. So that's something we'd like to explore more. Let's see. This is the big graphical takeaway from that work. We sampled 2 different bloom states. One was an active infection, but it was kind of at the peak of its bloom, whereas one was, saw a decrease in net primary productivity and a lot of virus-free in the water. We saw more oxylipins over here in this post-lytic state, and we saw 2 things. We saw stimulation of the surface ocean and particle-associated communities by these oxylipins. And so these are kind of in the vein of this like viral shunt, thinking of like DOC being remineralized and recycled and not exported. But we also saw some evidence that there was more export during these times. And so I would assert that this viral shunt versus viral shuttle, they're not really versus. I think they both occur at the same time. And I think the big question is like, what is the net impact in the carbon cycle? During viral infection, do you get more turnover in the surface ocean, but you get more export, and maybe, they cancel each other out. Or perhaps one of these wins over the other, but I think both of these processes are being stimulated by viral infection. It's kind of asking, which one is most of the carbon being partitioned into, could become the question. Checking our time. All right. We've done this with other viruses, but I wanted to talk about hatcheries really fast. I don't have too long, so I'll try to do it in 2 minutes. And I mentioned this because you all are so interested in abalone and oysters and other aquaculture production that I thought it was an interesting thing to at least talk about a little bit. A friend of mine who was working in Walpole, Maine at Mook Sea Labs, she had noticed that they're having these deformities they're noticing in their larvae. So this is what we're calling the cinnamon roll deformity. And these are what we're calling the duckbill deformity over here. And these are early warning signs of a slightly different problem where they're getting these digestive syndromes that were popping up. And so in 2019, they had a normal production year. So day 4 spat, so this is an oyster larvae. You can see that it just digested algae inside of its digestive gland. And by day 8, it's doubled in size. In 2020, Day 4, spat were ingesting food, but they weren't digesting the food. So you can see there's still a lot of structure. You can like almost see like the little squares that are the algae in those images. And in 4 days' time, they hadn't grown at all, and you can still see most of the intact algae. They did a ton of different experiments, and what they found was that when they removed UV pretreatment out of their intake into the hatchery, they didn't have the problem anymore. But when they did that, they ending up with a pathogen that slipped through because they weren't doing the UV filtration anymore. So we had to work on this problem a little bit more and figure out what compounds were actually, or what in the UV process was causing this to occur, and is there something other than just removing UV that we could do? They set up a bunch of experiments where they didn't add UV, and then they did add UV, and we sampled the lipidome in the plus UV and the minus UV treatments to see what was in there. And we observed a bunch of oxylipins being produced, as well as a bunch of pollutants. And so why would 1 micron filtered water release pollutants whenever you add UV? Why would they release oxylipins when you add UV? We're still working that out. But what we believe based on what we know about these types of compounds is that there must be some phytoplankton cells or some algal detritus or maybe even plant detritus that's getting through the 1 micron filter. And then when you add UV, you end up oxidizing those membrane lipids and releasing these oxylipins. We also think that based on some of the compounds that we identified in the UV-treated water, that microplastics might play a role. We also saw a bunch of pollutants, and so we think that maybe the microplastics, a thing they're known to do is to leach pollutants, organic pollutants specifically, and heavy metals onto them. And so then whenever we add UV, breaks them down and degrades them. Luckily, they found that if they let the water age for about 2-3 days after this UV treatment, these compounds must volatilize away, and you can grow the spat out. They're just fine. And we did try some experiments where we added oxylipins back in, and this is actually a precursor of the oxylipin. And so we add the oxylipin in, we add UV, and we get the same problem where they're ingesting, but they're not digesting the material. Versus at this time, they weren't having the problem anymore in the hatchery at all. And so over here, you could actually UV the water and it would be fine. It was only whenever you added the oxylipin back in that we get this recreation of this symptom. All right, so that brings me to the end of my talk. I hope I've shown you that there's lots of chemical signaling that's induced in the system through stress, be it viruses, be it silica stress, or UV even. And I hope I've also shown you that the types of oxylipins that are produced by a single species even can be really plastic depending on what the stress is. I've shown you that these types of compounds can impact dinoflagellate grazing behavior, and that they can also impact aquaculture operations here. I'd like to thank my co-authors, people at MOOC Farm have been really valuable in doing a lot of this work, as well as my PhD advisors lab at Woods Hole, and collaborators Kim and Kay at Rutgers and funding sources. Happy to take questions. Thank you.
We have time for 1 or 2 questions, maybe.
Thank you for the talk. So I don't know if you can answer it, but you're seeing all these big changes happening during the viral infection. And I was just overall, think between the different processes of virus making products, the organism making products in response versus just the organism falling apart and things leaking out, like, which of those processes playing the biggest role, you think, in the signatures?
Yeah, it's a good question. I think it has to do with like the cell to some layer or to some level falling apart. I think especially when we're seeing this difference, I mean, I think it's kind of all of it. It's like the virus infects, right? And then what is its mode of replication? Does it need to get inside of the nucleus so that it can, use all of the nuclear machinery? And then once, we see this change before we see a decrease in cell number. So they're still happily replicating for the most part. And then we start to see, this difference about 2 days before we see a decrease in cell.
That why like a DNA virus and RNA virus might give different signatures?
Yeah, I'm pretty sure that that's why they have different signatures. And we have a new system where we have 2 RNA viruses that infect at different times, or like have different time scales. One's like a 9-day infection, one's a 6-day infection. And so we're hoping to answer some of those questions more specifically. Like they're both RNA viruses in this case, so then we can maybe see, like, is it the same suite of compounds, just staggered in time, or is it like a completely different set of responses?
Thank you.
Please. Yeah, so lipidomics, I was telling John this earlier, we use like this omics term to apply to like genomics, transcriptomics, metabolomics. And the way I think of it is omics means that we're taking the entire pool of biomolecules and trying to annotate it. So in genomics, you're looking at all the genes in a sample and you're trying to annotate it. In lipidomics, you're looking at all the lipids in a sample and trying to annotate them and quantify as well. So that's really the crux of lipidomics, and then it really relies on mass spectrometry as a tool to look at the lipids. But yes. Yeah, so the question was about, in a warming climate, and, if you have more viral infection, does this become a bigger issue? I think the interesting thing with climate is that oxylipins are produced, I told you, these polyunsaturated fatty acids. You've got to have those to make these oxylipins. And those are predicted, the concentration of those is predicted to decrease with increasing temperatures. And so cells usually make a lot of EPA and DHA when they're trying to deal with cold, because cold will like kind of freeze up their membranes, and so they try to put membrane lipids in there that have lots of double bonds, and that makes it harder for or makes it easier for the membrane to be fluid. And so there's some papers that have modeled this, and they suggest there'll be like a 30% decrease in EPA and DHA in our ocean by 2100. So yeah, the problem becomes complicated, right? You would think it's more stressful, so there should be more of these oxylipins, but then we're changing temperature, so there's going to be less of these molecules available for organisms to produce them. It's a good question.
One last question.
Yeah. Yeah, annotation. It's great. A great question. So annotation generally means identifying. And so when we think about like genes, you would say like, okay, here's this sequence, this gene sequence, it matches this other gene sequence that we've seen in a database, and we know what that gene does. Whenever we're talking about like metabolites, you want to say like, okay, I think it's this metabolite. I broke it apart and it breaks apart like that metabolite would, so I'm pretty sure I'm going to call it that. So that's what we mean by annotation. You end up with like a big old list of like, okay, we saw like 10 different compounds, but maybe only 5 of them have really good IDs on them. So then you got to go do a little bit of legwork. Awesome. Thank you all so much. I really enjoyed visiting.